Hello, beautiful souls. Welcome to this quiet space, a place where minds slow down and hearts rest. Before we begin this gentle, sleepy journey, just a small moment of support. If this meditation brings you calm, comfort, or clarity, please subscribe to the channel, gently tap the like button, and leave a comment to let us know you are here. Your support helps us continue creating these peaceful spaces for your mind, for your heart. Now, there is nothing else you need to do, nothing to think about. Let's begin. Close your eyes now and slowly settle into a comfortable position. Take a gentle breath in and slowly breathe out. Let your shoulders relax, let your jaw loosen, and allow your whole body to feel heavy. Now, imagine we are beginning a quiet journey, not to outer space, not to deep oceans, but to a place much closer. Inside your mind, inside your heart, rests something very important, something that flows through your days, something that shapes your experiences, your emotions. Your emotions are natural. Your emotions are essential. They are ready to be felt. Tonight, we will travel through the quiet, gentle world inside your own mind, where emotions arise, where emotions are acknowledged, where emotions are gently, softly understood and balanced. Emotions are biological states that occur within the human body and brain in response to internal processes or external stimuli. These states involve patterns of neural activation, changes in hormonal levels, alterations in physiological functions, and gentle modifications in subjective experience. When we refer to emotions in scientific terms, we are describing coordinated responses that emerge from the integrated activity of multiple brain systems working together in ways that have been refined over millions of years of evolutionary time. The study of emotions involves examining how specific regions of the brain communicate with one another, how chemical messengers travel between neurons, and how the body responds to signals originating in the central nervous system. Emotions are not simply feelings or thoughts, but rather they represent complex biological phenomena that involve the coordination of neural circuits, endocrine responses, autonomic nervous system activity, and behavioral tendencies that all work together slowly to produce what we experience as emotional states. The brain processes information continuously, receiving input from sensory organs, integrating that information with stored memories and current goals, and then generating appropriate responses. Within this ongoing stream of processing, emotions emerge as particular patterns of activation that help to organize behavior and gently direct attention towards stimuli that may be relevant for survival, reproduction, or social functioning. These patterns involve changes in how quickly neurons fire, which neurochemical systems become active, and how different brain regions synchronize their activity with one another. The scientific understanding of emotions has developed slowly over many decades, with researchers gradually mapping out the neural pathways involved, identifying the chemical messengers that modulate emotional responses, and documenting how these systems develop across the lifespan and vary across different contexts and individuals. When scientists study emotions, they often begin by distinguishing between several basic emotional states that appear to be relatively universal across human populations. These states include responses such as contentment, displeasure, interest, and calmness, among others. Each of these states corresponds to particular patterns of brain activity that can be measured using various neuroimaging techniques, such as functional magnetic resonance imaging, electroencephalography, and positron emission tomography. These techniques allow researchers to observe which brain regions become more active during different emotional states, how quickly these activations occur, and how they spread throughout the brain over time. By examining these patterns across many individuals and many situations, scientists have slowly built up a detailed picture of the neural architecture that supports emotional experience. The evolutionary origins of emotions extend back millions of years into the deep history of vertebrate life. Early vertebrate species possessed basic neural structures that allowed them to respond to their environment in adaptive ways. Approaching stimuli that signaled potential resources and withdrawing from stimuli that signaled potential harm. These primitive response systems gradually became more sophisticated as vertebrate brains evolved, with new layers of neural tissue being added on top of older structures, creating increasingly complex mechanisms for evaluating stimuli and selecting appropriate responses. The emotional systems we observe in modern humans represent the culmination of this long evolutionary process, incorporating ancient mechanisms that we share with other mammals, other vertebrates, and even more distantly related species, while also including newer mechanisms that emerged more recently in primate evolution and in the human lineage, specifically. In the earliest vertebrates, basic approach and withdrawal behaviors were controlled by relatively simple neural circuits located in the brainstem and other subcortical structures. These circuits allowed organisms to move toward food sources, potential mates, and safe environments, while moving away from predators, toxins, and other threats. Over evolutionary time, as brains became larger and more complex, additional neural structures evolved that could process information in more sophisticated ways, allowing for more nuanced evaluations of environmental stimuli and more flexible behavioral responses. The development of the limbic system in early mammals represented an important step in this evolutionary process, as this collection of interconnected brain structures provided enhanced capabilities for processing social information, forming memories, and gently modulating basic approach and withdrawal responses based on learning and experience. The limbic system includes several structures that play crucial roles in emotional processing, including the amygdala, the hippocampus, the hypothalamus, and portions of the thalamus, among others. These structures are interconnected through dense networks of neural fibers that allow information to flow between them, creating feedback loops and parallel processing pathways that contribute to the complexity of emotional experience. The evolution of the limbic system allowed mammals to develop more sophisticated social behaviors, stronger bonds between parents and offspring, and greater flexibility in responding to environmental challenges. As primate brains continued to evolve, additional developments occurred in cortical regions, particularly in the frontal lobes, that provided even greater capacity for regulating emotional responses, anticipating future events, and integrating emotional information with complex cognitive processes. The human brain retains all of these evolutionarily older structures and mechanisms, layered together with more recently evolved cortical systems that allow for conscious reflection on emotional states, voluntary regulation of emotional responses, and the integration of emotional information into complex decision-making processes. This layered evolutionary architecture means that human emotional responses involve the coordinated activity of both ancient subcortical structures and newer cortical regions, with information flowing back and forth between these different levels of organization in ways that shape our moment-to-moment emotional experience. Understanding emotions from an evolutionary perspective helps us recognize that these responses are not arbitrary or irrational, but rather reflect deeply ingrained biological mechanisms that have been shaped by natural selection to serve important adaptive functions. The amygdala is a small, almond-shaped structure located deep within the temporal lobes of the brain, with one amygdala present in each hemisphere. This structure plays a particularly important role in processing emotionally salient information, especially information related to potential threats or rewards in the environment. The amygdala receives input from many different brain regions, including sensory cortices that process visual, auditory, and other types of information, as well as from structures involved in memory, such as the hippocampus. When the amygdala receives information indicating that a stimulus may be emotionally significant. It generates outputs that influence many other brain regions and body systems, helping to coordinate an appropriate emotional response. These outputs travel to regions involved in attention, memory, physiological arousal, and behavioral action, creating a coordinated pattern of changes throughout the brain and body. The amygdala is not a single, uniform structure, but rather consists of several distinct nuclei, each with somewhat different patterns of connectivity and functional properties. The lateral nucleus of the amygdala receives sensory information from cortical and subcortical sources and begins the process of evaluating whether incoming stimuli have emotional significance. Information then flows to the basal nucleus and eventually to the central nucleus, which serves as a major output station, sending projections to many different brain regions, including the hypothalamus brainstem nuclei that control physiological responses, and cortical regions involved in conscious perception and decision-making. This flow of information through the amygdala occurs very rapidly, often within a few hundred milliseconds of stimulus presentation, allowing for quick emotional responses that can occur before conscious awareness of the stimulus has fully developed. Research conducted over several decades has shown that damage to the amygdala can impair the ability to recognize emotionally significant stimuli, particularly those related to potential threats. Studies using neuroimaging techniques have demonstrated that the amygdala responds to a wide range of emotionally relevant stimuli, including images, sounds, and even abstract symbols that have acquired emotional significance through learning. The amygdala also plays an important role in emotional memory formation, helping to strengthen memories for events that occurred during emotionally arousing states. This function helps explain why we tend to remember emotionally significant events more vividly than neutral events, a phenomenon that has clear adaptive value, as it allows organisms to learn from past experiences and adjust their behavior accordingly. The hippocampus is another structure within the limbic system that plays a crucial role in emotional processing. Though its primary function relates to memory formation and spatial navigation, the hippocampus is located adjacent to the amygdala in the medial temporal lobe, and these two structures are extensively interconnected, allowing for close coordination between emotional processing and memory formation. The hippocampus receives information about ongoing events and experiences, and processes this information in ways that allow it to be stored as long-term memories that can be retrieved later. When emotionally significant events occur, signals from the amygdala influence hippocampal processing in ways that tend to strengthen the resulting memories, making them more stable and more easily retrieved. The hippocampus contains specialized types of neurons that are particularly well suited for encoding information about experiences and contexts. These neurons show plasticity in their connections, meaning that the strength of synaptic connections between neurons can be modified based on patterns of activity, providing a biological mechanism for learning and memory. The interaction between the hippocampus and the amygdala during emotional events creates particularly strong memories that link together information about what happened, where it happened, and what emotional significance the event held. These rich, contextualized memories help guide future behavior by allowing individuals to recognize when they are in situations similar to those they have encountered before, and to anticipate likely outcomes based on past experience. Damage to the hippocampus can result in severe memory impairments, including difficulty forming new, long-term memories, while leaving older memories relatively intact. Studies of individuals with hippocampal damage have provided valuable insights into the role this structure plays in memory and emotion. These studies reveal that while the hippocampus is essential for forming new explicit memories, the ability to experience emotional responses in the moment may remain relatively preserved, suggesting that immediate emotional reactions can be generated without requiring intact hippocampal function. Though the ability to learn from emotional experiences over time depends critically on hippocampal processing. The prefrontal cortex represents a more recently evolved portion of the brain that plays essential roles in regulating emotional responses, planning future actions, and integrating emotional information with cognitive processes. The prefrontal cortex is located in the frontal portion of the brain, just behind the forehead, and it is particularly well developed in primates, especially in humans. This region can be divided into several subregions, each with somewhat different patterns of connectivity and functional specialization. The ventromedial prefrontal cortex, located on the underside of the frontal lobes, is particularly important for processing information about rewards and punishments, and for integrating emotional information into decision-making processes. The dorsolateral prefrontal cortex, located on the outer surface of the frontal lobes, is more involved in working memory, planning, and cognitive control, including the voluntary regulation of emotional responses. The prefrontal cortex receives input from many other brain regions, including sensory cortices, limbic structures such as the amygdala and hippocampus, and other cortical regions involved in various cognitive functions. This rich pattern of connectivity allows the prefrontal cortex to integrate information from many sources and to exert regulatory control over other brain systems. Connections from the prefrontal cortex to the amygdala, for example, allow the prefrontal cortex to modulate amygdala activity, potentially dampening emotional responses when they might be excessive or inappropriate for the current context. This regulatory function develops gradually across childhood and adolescence. As the prefrontal cortex undergoes an extended period of maturation, that continues into the third decade of life. Studies examining the effects of prefrontal cortex damage have revealed important insights into the role this region plays in emotional regulation and social behavior. Individuals with damage to ventromedial regions of the prefrontal cortex may show impaired decision-making, particularly in situations involving uncertainty or long-term consequences. Even when their basic intellectual abilities remain intact, they may also show altered emotional responses, sometimes appearing less emotionally reactive than would be expected in particular situations, or alternatively, showing emotional responses that seem inappropriate for the social context. These findings highlight the importance of the prefrontal cortex in linking emotional responses to contextual information and long-term goals, allowing for flexible and adaptive behavior across diverse situations. The hypothalamus is a small structure located at the base of the brain, just above the brainstem, that serves as a crucial link between the nervous system and the endocrine system. Despite its small size, the hypothalamus plays essential roles in regulating many basic physiological functions, including body temperature, hunger, thirst, sleep-wake cycles, and hormonal responses. The hypothalamus also plays important roles in emotional responses. By controlling autonomic nervous system activity and by triggering the release of hormones from the pituitary gland. When emotional responses are initiated, often through amygdala activity, the hypothalamus receives signals that lead to coordinated changes in body state, including alterations in heart rate, blood pressure, respiration, and the release of stress hormones such as cortisol. The hypothalamus contains several distinct nuclei, each involved in different regulatory functions. The paraventricular nucleus, for example, contains neurons that release corticotropin-releasing hormone, which initiates a cascade of hormonal responses that ultimately lead to the release of cortisol from the adrenal glands. This hormonal response helps mobilize energy resources and gently modulate various body systems in ways that support responses to challenging situations. Other hypothalamic nuclei are involved in regulating food intake, sexual behavior, and other basic, motivated behaviors that are closely linked to emotional states. The hypothalamus also contains neurons that project to brainstem regions, controlling autonomic nervous system activity, allowing it to rapidly influence heart rate, blood pressure, digestion, and other physiological processes. The close interconnection between the hypothalamus and other limbic structures creates integrated systems for responding to emotional stimuli. When the amygdala detects an emotionally significant stimulus, it sends signals to the hypothalamus that trigger appropriate physiological responses. These responses occur rapidly and largely automatically, without requiring conscious deliberation, allowing the body to prepare for action before conscious awareness of the situation has fully developed. At the same time, feedback from the body to the brain, including information about heart rate, breathing, and hormonal state, influences ongoing emotional processing, creating loops of interaction between brain and body that gently shape emotional experience. Neurotransmitters are chemical messengers that allow neurons to communicate with one another across the small gaps called synapses that separate them. Different types of neurotransmitters have different effects on the neurons they interact with, and the complex interplay of multiple neurotransmitter systems helps determine patterns of brain activity and consequently emotional states. Several neurotransmitters have been identified as particularly important for emotional processing, including dopamine, serotonin, norepinephrine, and various neuropeptides such as oxytocin and corticotropin-releasing hormone. Each of these chemical messengers is produced by specific populations of neurons and released in particular patterns in response to various types of stimulation, creating complex and dynamic neurochemical environments that gently modulate emotional responses. Dopamine is a neurotransmitter that has been extensively studied for its role in motivation, reward processing, and certain aspects of emotional experience. Dopamine is produced by neurons located in several brainstem nuclei, particularly the substantia nigra and the ventral tegmental area. These neurons send projections to many brain regions, including the striatum, prefrontal cortex, amygdala, and hippocampus, allowing dopamine to influence a wide range of neural processes. Dopamine release increases in response to rewarding stimuli or cues that predict rewards, and this release appears to play a role in reinforcement learning by strengthening connections between neurons that were active when the reward occurred. This mechanism helps organisms learn which actions and situations tend to lead to positive outcomes, shaping behavior slowly over time in adaptive ways. The role of dopamine in emotional processing is complex and extends beyond simple pleasure or reward. Dopamine signaling is involved in motivated behavior more generally, helping to energize behavior and gently direct attention toward goals. Variations in dopamine function have been linked to individual differences in personality traits related to approach behavior, novelty seeking, and sensitivity to rewards. Disruptions in dopamine systems have been implicated in various psychiatric conditions, including depression, where reduced dopamine signaling may contribute to decreased motivation and diminished experience of pleasure, and in conditions involving excessive approach behavior or impulsivity. The dopamine system interacts closely with other neurotransmitter systems and with the broader neural circuits involved in emotional processing, contributing to the overall pattern of emotional responding. Serotonin is another neurotransmitter that plays important roles in emotional processing and regulation. Serotonin is produced by neurons located in the Ravi nuclei of the brainstem, which send widespread projections throughout the brain and spinal cord, allowing serotonin to influence many different neural systems. Serotonin has been implicated in the regulation of mood, with reduced serotonin function often associated with low mood states. Though the relationship between serotonin and emotion is complex and not fully understood, Serotonin signaling influences many processes relevant to emotional experience, including sleep-wake cycles, appetite, pain sensitivity, and the regulation of other neurotransmitter systems. One important function of serotonin appears to be the modulation of responses to aversive or challenging situations. Serotonin signaling can influence how strongly individuals respond to negative outcomes or stressful experiences. With adequate serotonin function helping to maintain emotional stability in the face of difficulties, the serotonin system shows plasticity in response to chronic stress or other sustained challenges, with prolonged stress sometimes leading to alterations in serotonin synthesis, release, or receptor sensitivity. These changes may contribute to the development of persistent alterations in mood or emotional reactivity. Many medications used to treat mood-related conditions work by modifying serotonin signaling, often by preventing the reuptake of serotonin from synapses, thereby increasing the amount of serotonin available to interact with receptors. Norepinephrine, also known as noradrenaline, is a neurotransmitter that plays important roles in arousal, attention, and responses to challenging situations. Norepinephrine is produced by neurons located in the locus coeruleus, a small nucleus in the brainstem that sends projections throughout the brain. When the locus coeruleus becomes active, norepinephrine is released broadly across many brain regions, increasing alertness and focusing attention on salient stimuli. This widespread release of norepinephrine is part of the arousal response that occurs during emotionally significant events, helping to coordinate brain-wide changes in processing that support rapid responding to important situations. The norepinephrine system is closely linked to the body's stress response systems. When potentially threatening or challenging situations are detected, norepinephrine release increases in the brain, while the related molecule, epinephrine or adrenaline, is released into the bloodstream from the adrenal glands. These coordinated changes help mobilize energy resources, increase cardiovascular activity, and enhance sensory processing and attention. The norepinephrine system interacts with other neurotransmitter systems, including the serotonin system and the dopamine system, and with the various brain regions involved in emotional processing. Variations in norepinephrine function have been linked to individual differences in emotional reactivity and to various conditions involving altered emotional responses. Oxytocin is a neuropeptide, a type of chemical messenger composed of a short chain of amino acids that has received considerable research attention for its role in social behavior and bonding. Oxytocin is produced by neurons in the hypothalamus and is released both within the brain, where it acts as a neurotransmitter, and into the bloodstream, where it acts as a hormone. Oxytocin has been implicated in a variety of social and emotional processes, including the formation of social bonds, the experience of trust, and various aspects of parental and reproductive behavior. The oxytocin system shows increased activity during positive social interactions, and experimental administration of oxytocin has been found to influence social behavior in complex ways that depend on context and individual characteristics. The role of oxytocin in emotional processing appears to involve modulation of activity in brain regions such as the amygdala and prefrontal cortex. Oxytocin can influence how individuals process social information, potentially reducing reactivity to social threats, while enhancing attention to social cues and positive social stimuli. The oxytocin system shows plasticity in response to social experiences, with repeated positive social interactions potentially leading to changes in oxytocin receptor density or signaling that could influence future social and emotional responses. The oxytocin system represents one example of how neuropeptide systems contribute to the complex neurochemical environment that supports emotional experience, working alongside the classical neurotransmitter systems to modulate neural activity in ways that shape behavior and subjective experience. Cortisol is a steroid hormone released from the adrenal glands in response to stress and plays important roles in mobilizing energy resources and modulating various physiological systems. Cortisol release is controlled by a system known as the hypothalamic pituitary adrenal axis. When emotionally challenging situations are encountered, the hypothalamus releases corticotropin-releasing hormone, which travels to the pituitary gland and triggers the release of adrenocorticotropic hormone into the bloodstream. This hormone then stimulates the adrenal glands to release cortisol. Cortisol has widespread effects throughout the body and brain, influencing metabolism, immune function, and neural activity in various brain regions. Cortisol can influence memory formation with moderate levels of cortisol during or shortly after an event, tending to strengthen memories for that event, while very high or chronic elevations of cortisol can have different effects on memory and neural function. The cortisol system shows a natural daily rhythm, with levels typically highest in the morning and gradually declining throughout the day, reaching their lowest point during the early night. This rhythm is influenced by sleep-wake patterns and by light exposure, representing one of many circadian rhythms that organize physiological function across the 24-hour day. Emotional experiences can cause increases in cortisol that override this baseline rhythm, and chronic stress can lead to alterations in the normal cortisol pattern. The cortisol system interacts with neurotransmitter systems in the brain and with other hormonal systems, contributing to the complex web of biological mechanisms that support emotional responses and regulate bodily function. The processing of emotional information in the brain occurs through multiple parallel pathways that work simultaneously to evaluate stimuli, activate appropriate response systems, and generate conscious emotional experience. When a stimulus is encountered, sensory information first reaches primary sensory cortices, where basic features of the stimulus are extracted. This information then flows to higher-level sensory areas, where more complex patterns are identified, and simultaneously to limbic structures, such as the amygdala, where the emotional significance of the stimulus is rapidly evaluated. The amygdala can begin to respond to emotional stimuli very quickly, within approximately 100 to 200 milliseconds of stimulus presentation. Before complete conscious processing of the stimulus has occurred, this rapid response pathway allows for quick reactions to potentially important stimuli, which may have provided survival advantages. In evolutionary contexts where rapid responding to threats was essential, at the same time that this rapid subcortical processing is occurring, Information about the stimulus is also being processed through cortical pathways that provide more detailed and nuanced analysis. Visual information, for example, follows a pathway from the retina through the thalamus to the primary visual cortex, and then through a series of higher visual areas that extract increasingly complex features and ultimately allow for conscious perception and recognition of visual objects. This cortical processing takes longer than the rapid subcortical pathway, but provides more detailed information that can then be integrated with the initial emotional evaluation. Information from sensory cortices reaches the prefrontal cortex, where it can be integrated with memories, goals, and contextual information to generate more sophisticated evaluations of the situation and to regulate emotional responses if needed. The interaction between subcortical and cortical processing pathways creates a dynamic system where rapid emotional responses can be initiated quickly, while also allowing for more deliberative processing that can modulate or override initial reactions when appropriate. The prefrontal cortex can send signals to the amygdala that modulate its activity, potentially dampening emotional responses when they are not appropriate for the current context, or when longer-term goals would be better served by restraint. This regulatory capacity depends on the development and integrity of connections between prefrontal cortex and limbic structures. And these connections continue to mature throughout childhood and adolescence, which may help explain why emotional regulation abilities tend to improve gradually with age during development. After initial evaluation of emotional significance, various response systems are activated to produce coordinated emotional responses. The amygdala sends outputs to the hypothalamus, which triggers autonomic nervous system responses and hormonal changes. Outputs to brainstem motor nuclei influence facial expressions and body postures. Outputs to cortical regions influence attention, perception, and decision-making. Outputs to memory systems influence how the current experience will be stored for future reference. All of these changes occur in parallel, creating a coordinated pattern of neural, physiological, behavioral, and experiential changes that constitute an emotional response. The specific pattern of changes depends on the nature of the eliciting stimulus, the current context, the individual's past experiences, and their current physiological and psychological state. The distinction between emotions, feelings, and moods represents an important conceptual framework for understanding different aspects of affective experience. Emotions, in the scientific usage of the term, refer to relatively brief episodes of coordinated changes in brain and body state that occur in response to specific eliciting events. These episodes typically last from seconds to minutes and involve the kinds of neural, physiological, and behavioral changes that have been described in previous sections. Feelings refer to the subjective, conscious experience of emotional states, the internal sensations and mental qualities that accompany emotional episodes, while emotions involve objective, measurable changes in brain and body state. Feelings are inherently subjective and can only be known directly by the individual experiencing them, though they can be described and communicated to others through language. The relationship between emotions and feelings has been a subject of considerable theoretical debate in psychology and neuroscience. Some theories propose that feelings arise directly from the conscious perception of bodily changes that occur during emotional episodes, suggesting that we feel a particular way because we become aware of changes in heart rate, breathing, facial expression, and other physiological parameters. Other theories propose that feelings arise more directly from central brain processes, with bodily changes being parallel consequences of emotional brain states, rather than the primary source of feelings. Current evidence suggests that both bodily feedback and central brain processes contribute to the generation of feelings, with conscious emotional experience emerging from the integration of information about internal body states, with cognitive appraisals of the eliciting situation and its significance. Moods differ from emotions in their duration and in the specificity of their eliciting causes, while emotions are relatively brief responses to specific events. Moods are more sustained states that can last for hours or days, and that often lack clearly identified identifiable, triggering events. Moods influence how individuals respond to events and information they encounter, biasing perception, thought, and behavior in ways that are consistent with the mood state. During positive mood states, individuals may be more likely to notice positive aspects of their environment, to interpret ambiguous situations favorably, and to engage in approach behaviors. During negative mood states, attention may be biased toward potential problems or threats. Ambiguous situations may be interpreted more negatively, and individuals may be less motivated to engage in effortful activities. The neural mechanisms underlying moods involve many of the same brain regions and neurotransmitter systems that support emotions. But moods may involve more sustained changes in neural activity patterns or in the sensitivity of particular neural systems. Changes in neurotransmitter function, such as alterations in serotonin or dopamine signaling, may contribute to mood states and may help explain why moods can persist even in the absence of ongoing emotional provocations. Environmental factors such as sleep quality, nutrition, physical activity, social interactions, and exposure to natural light can all influence mood states, suggesting that moods reflect the current operating state of brain systems that are sensitive to a wide range of internal and external factors. Emotional regulation refers to the processes by which individuals influence which emotions they have, when they have them, and how they experience and express them. Emotional regulation can occur through various mechanisms, operating at different points in the emotion generation process. One form of emotional regulation involves situation selection, where individuals choose to approach or avoid situations based on the emotions those situations are likely to elicit. Another form involves situation modification, where individuals take actions to change aspects of a situation to alter its emotional impact. Attentional deployment involves directing attention toward or away from emotionally relevant aspects of a situation, which can influence the intensity of emotional responses. Cognitive change involves altering how one thinks about a situation to change its emotional significance, such as through reappraisal processes, where situations are reinterpreted in ways that alter their meaning. Response modulation involves influencing emotional responses after they have already been initiated, such as through suppression of emotional expressions, or through techniques to calm physiological arousal. Different regulatory strategies have different effects on emotional experience and on physiological responses, with some strategies being generally more effective or less costly than others. Cognitive reappraisal, for example, where individuals reinterpret situations in ways that alter their emotional significance, has been found to effectively reduce subjective emotional experience and physiological arousal without the costs associated with suppression, which involves inhibiting emotional expressions after emotional responses have already been generated, and which tends to reduce behavioral expression while leaving physiological arousal unchanged or even increased. The neuromechanisms supporting emotional regulation involve interactions between prefrontal cortex regions involved in cognitive control and limbic regions involved in generating emotional responses. When individuals engage in cognitive reappraisal, for example, neuroimaging studies show increased activity in lateral and medial prefrontal cortex regions, along with decreased activity in the amygdala, consistent with the idea that prefrontal regions are exerting regulatory control over limbic emotional responses. The capacity for emotional regulation develops gradually across childhood and adolescence as prefrontal cortex regions mature and as individuals gain experience with different regulatory strategies and learn which strategies are effective in different contexts. Individual differences in emotional regulation capacity have been linked to various aspects of psychological health and social functioning. Individuals who are able to effectively regulate their emotions tend to experience better psychological well-being, have more satisfying social relationships, and show better physical health outcomes. Difficulties with emotional regulation have been implicated in various psychological conditions, including mood disorders, where individuals may experience emotions that are excessively intense or prolonged, and various behavioral conditions where difficulties regulating emotions may contribute to impulsive or maladaptive behaviors. Understanding the mechanisms of emotional regulation and how these mechanisms can be strengthened has become an important focus of research aimed at developing interventions to support psychological health. Emotions play important roles in decision-making processes, influencing choices in ways that can be adaptive, but that can also sometimes lead to biases or suboptimal decisions. When individuals are making decisions, they often experience emotional responses to the different options they are considering and to the anticipated outcomes of those options. These emotional responses can serve as sources of information that guide choice, with options that elicit positive emotional responses being favored over those that elicit negative responses. This process can allow for rapid decision-making in complex situations where fully analyzing all relevant information would be time-consuming or impossible, as emotional responses can summarize past experiences and learned associations in ways that guide behavior without requiring explicit deliberation. The somatic marker hypothesis, proposed by neuroscientist Antonio Damasio, suggests that emotional signals from the body play an essential role in guiding decision-making, particularly in complex situations involving uncertainty and multiple conflicting considerations. According to this theory, when individuals contemplate different possible actions, they experience subtle emotional responses or somatic markers that reflect the predicted outcomes of those actions based on past experience. These somatic markers help to highlight options that have led to positive outcomes in the past, while steering individuals away from options that have led to negative outcomes. Without these emotional signals, decision-making becomes much more difficult and may be impaired, as individuals lack an intuitive sense of which options are likely to be beneficial. Research examining individuals with damage to ventromedial prefrontal cortex regions involved in processing emotional information has provided support for the importance of emotions in decision-making. These individuals often show preserved intellectual abilities and can reason logically about the pros and cons of different options. When these are presented abstractly, but they show marked impairments in real-world decision-making, particularly in personal and social domains. They may make choices that lead to negative consequences for themselves and others, apparently because they lack the emotional signals that would normally guide them toward advantageous options. These findings suggest that far from being opposed to rationality, emotions constitute an essential component of effective decision-making in complex, real-world contexts. At the same time, emotional influences on decision-making can sometimes lead to biases or errors. Strong emotional states can narrow attention and lead to overweighting of information consistent with the current emotional state, while underweighting contradictory information. Immediate emotional responses can sometimes lead individuals to favor short-term rewards over larger, long-term benefits, or to avoid options that elicit negative emotions, even when those options might be objectively better choices. Understanding both the adaptive functions of emotional influences and the potential biases they can introduce is important for developing a complete picture of how humans make choices in various contexts. Emotional development occurs across the entire human lifespan, with emotional responses, regulation abilities, and the neural systems supporting emotion, all showing important changes from infancy through old age. Infants are born with the capacity to experience and express basic emotional states, showing different patterns of facial expressions, vocalizations, and body movements in response to different types of stimulation. These early emotional expressions serve important communicative functions, allowing infants to signal their needs and states to caregivers. Over the first years of life, emotional expressions become more differentiated, and children develop increasing awareness of their own emotional states and the emotional states of others. Language development supports emotional development by providing children with words to label and describe their feelings, which may facilitate emotional awareness and regulation during childhood. Important developments occur in the neural systems supporting emotion and in children's abilities to regulate their emotional responses. The prefrontal cortex undergoes extensive development during childhood and adolescence, and connections between prefrontal regions and limbic structures are gradually refined. These neural changes support improvements in emotional regulation abilities, with children showing gradually increasing capacity to modulate their emotional responses in accordance with social rules and personal goals. Children also develop more sophisticated understanding of emotions, learning about the causes and consequences of different emotional states, and about the fact that people can feel different emotions in the same situation depending on their perspective and personal concerns. Adolescence represents a period of particularly important changes in emotional functioning. The onset of puberty brings hormonal changes that influence emotional reactivity, and brain imaging studies have revealed that limbic regions involved in emotional processing undergo important changes during adolescence, while prefrontal regions involved in cognitive control continue to mature. This asynchrony in development has been proposed to contribute to the increased emotional reactivity and sometimes greater difficulty with emotion regulation that is often observed during adolescence. At the same time, adolescents are navigating increasing social complexity and facing important developmental tasks related to identity formation and increasing independence, all of which occur in contexts where emotional experiences are particularly salient. During adulthood, emotional functioning shows both stability and change. Personality traits related to emotional functioning tend to be relatively stable across adulthood, with individual differences in emotional reactivity and regulation showing consistency over time. At the same time, there is evidence for gradual changes In average emotional experience across adulthood, with middle-aged and older adults often reporting greater emotional well-being than younger adults, despite facing various challenges and losses. Older adults also show some differences in emotional processing and regulation compared to younger adults, sometimes placing greater emphasis on emotion regulation goals and showing enhanced ability to avoid or disengage from emotionally negative information while maintaining attention to positive emotional material. These age-related changes in emotional experience and regulation have been interpreted in various ways. One perspective emphasizes that as individuals age and perceive their remaining time as more limited, their goals and priorities may shift in ways that lead to greater emphasis on emotionally meaningful experiences and relationships, and less concern with information or activities that lack immediate emotional significance. Neural changes that occur with aging, including gradual changes in prefrontal cortex function and in neurotransmitter systems, may also contribute to age-related differences in emotional processing. Understanding emotional development across the full lifespan requires considering the complex interplay of neural changes, life experiences, changing goals and priorities, and the various social and environmental contexts in which people live at different ages. The relationship between emotions and sleep represents an important area of inquiry that has implications for understanding both emotional functioning and sleep processes during waking hours. Emotional experiences and the regulation of those experiences can influence sleep, with emotional arousal and difficulty regulating emotions potentially interfering with sleep onset and sleep quality. Conversely, sleep has important effects on emotional functioning, with sleep deprivation or poor sleep quality leading to alterations in emotional reactivity and regulation during subsequent waking periods. Research has shown that even modest sleep restriction can lead to increased emotional reactivity to negative stimuli and reduced effectiveness of emotion regulation strategies, suggesting that adequate sleep is important for optimal emotional functioning. During sleep itself, emotional processing continues to occur. The brain does not simply shut down during sleep, but rather shows organized patterns of activity that differ across different sleep stages. During rapid eye movement sleep, which is the sleep stage most strongly associated with vivid dreaming, brain activity patterns include activation of limbic regions, such as the amygdala and hippocampus, while prefrontal cortex regions show reduced activity compared to waking. This pattern of activity during rapid eye movement sleep may allow for emotional memories to be processed and integrated in the absence of prefrontal regulatory control, potentially serving functions related to emotional memory consolidation and emotional adaptation. The experience of emotions during dreams can be intense and may include emotional states and responses that differ from typical waking emotional experiences. Non-rapid eye movement sleep, which occupies the majority of sleep time and includes deeper sleep stages, is characterized by different patterns of brain activity, including synchronized slow oscillations that propagate across cortical regions. During these deeper sleep stages, autonomic nervous system activity is generally reduced and stabilized, with heart rate and breathing becoming more regular and blood pressure decreasing. The processing of emotional memories may continue during non-rapid eye movement sleep, with memories being reactivated and gradually integrated into existing knowledge structures. Some research suggests that the different sleep stages may play complementary roles in emotional processing, with rapid eye movement sleep being particularly important for processing emotionally salient aspects of memories, while non-rapid eye movement sleep supports the integration of new emotional memories with existing knowledge. The autonomic nervous system serves as a crucial link between emotional brain processes and bodily responses, translating emotional states into physiological changes throughout the body. The autonomic nervous system consists of two main branches: the sympathetic and parasympathetic nervous systems, which work together to regulate the function of organs, and systems throughout the body, including the heart, lungs, digestive system, and various glands. The sympathetic nervous system generally activates responses that mobilize energy and prepare the body for action, increasing heart rate, dilating airways in the lungs, and redirecting blood flow toward muscles and away from digestive processes. The parasympathetic nervous system generally supports restorative processes, slowing heart rate, supporting digestion, and promoting energy conservation during emotional responses, particularly those involving high arousal or activation. Sympathetic nervous system activity increases, leading to the various physiological changes that have been described. These changes occur largely automatically, without requiring conscious effort or awareness, though individuals can become aware of these changes through interoception, the sensing of internal body states, the experience of becoming aware of increased heart rate, rapid breathing, or other physiological changes can itself influence emotional experience, contributing to the overall feeling of being in a particular emotional state. The relationship between autonomic nervous system activity and emotional experience is bidirectional, with emotional states influencing autonomic activity and autonomic activity influencing the ongoing experience of emotion. Individual differences in autonomic nervous system function have been linked to differences in emotional reactivity and regulation. Some individuals show greater autonomic reactivity with larger changes in heart rate, blood pressure, and other parameters in response to emotional stimuli, while others show more modest responses. These individual differences may reflect genetic factors, early developmental experiences, current health status, and habitual emotional regulation strategies. Individuals who engage in regular practices, such as physical exercise or certain types of meditation, may show alterations in resting autonomic tone and in reactivity to emotional stimuli, suggesting that autonomic function shows plasticity in response to behavior and experience. The measurement of autonomic nervous system activity provides researchers with objective indicators of emotional responses that can complement self-reported emotional experiences. Heart rate variability, electrodermal activity, respiration rate, and blood pressure can all be measured non-invasively and provide information about autonomic responses occurring during emotional episodes. Heart rate variability, which refers to the variation in time intervals between successive heartbeats, has received particular research attention as an indicator of parasympathetic nervous system influence on the heart, and as a potential marker of emotional regulation capacity. Higher resting heart rate variability is generally associated with better emotional regulation and greater behavioral flexibility, while reduced heart rate variability has been observed in various conditions involving emotional dysregulation. Emotions influence cardiovascular function through multiple mechanisms involving both neural and hormonal pathways. The heart receives input from both sympathetic and parasympathetic branches of the autonomic nervous system, which influence heart rate and the force of cardiac contractions. During states of high arousal or activation, sympathetic influence increases, while parasympathetic influence decreases, leading to increases in heart rate and cardiac output. Hormonal signals, including epinephrine, released from the adrenal glands, also influence cardiac function, reinforcing the effects of direct neural input. These coordinated changes in cardiovascular function support the metabolic demands of emotional responding, ensuring adequate oxygen and nutrient delivery to tissues throughout the body. The effects of emotions on breathing are similarly coordinated with other physiological changes. Breathing rate and depth are influenced by both voluntary control and automatic regulation, with the brainstem containing neural circuits that generate the basic rhythm of respiration during emotional responses. particularly those involving high arousal. Breathing patterns change in ways that increase oxygen uptake and carbon dioxide removal. The experience of altered breathing patterns can be quite noticeable and can contribute significantly to the overall subjective experience of particular emotional states. Individuals experiencing certain emotional states may notice their breathing becoming rapid and shallow, or may experience sensations of breathlessness or tightness in the chest. These respiratory changes are coordinated with cardiovascular changes and with alterations in other body systems to support appropriate responses to emotional situations. Digestive processes are also influenced by emotional states through autonomic nervous system pathways. The digestive system receives extensive innervation from both sympathetic and parasympathetic branches of the autonomic nervous system, which regulate various aspects of digestive function, including the motility of the digestive tract, the secretion of digestive enzymes and fluids, and blood flow to digestive organs. During states of high arousal or activation, sympathetic nervous system activity generally inhibits digestive processes, as energy and resources are redirected towards systems more immediately relevant for responding to challenges. This inhibition of digestion can manifest in various ways, including reduced appetite, slowed movement of material through the digestive tract, and alterations in digestive comfort with sustained or repeated emotional arousal. These effects on digestive function can accumulate and potentially contribute to various digestive difficulties. The bidirectional communication between the brain and the digestive system occurs not only through autonomic nervous system pathways, but also through hormonal signals and through the enteric nervous system, a complex network of neurons embedded in the walls of the digestive tract. This enteric nervous system can function somewhat independently of the central nervous system, but is also extensively connected to the brain through the vagus nerve and other pathways. Signals from the digestive system can influence brain function, and emotional states. While emotional states and stress can influence digestive processes, creating complex loops of interaction between these systems. Understanding these gut-brain interactions has become an active area of research, with implications for understanding various aspects of emotional experience and regulation. When examining emotions from a cross-cultural perspective, researchers have documented both universal and culturally variable aspects of emotional experience and expression. Certain basic patterns of emotional expression, particularly facial expressions, show remarkable consistency across different cultures, suggesting that these expressions have deep evolutionary roots and represent universal aspects of human emotional communication. At the same time, there is substantial cultural variation in many aspects of emotional experience and expression, including the situations that elicit particular emotions, the intensity with which emotions are expressed, the appropriateness of expressing different emotions in different contexts, and the words and concepts used to describe and categorize emotional experiences. Research conducted across diverse cultural contexts has revealed that while people in different cultures generally agree on the emotional meaning of certain facial expressions, there are differences in how frequently different expressions are displayed, how intensely they are expressed, and how they are interpreted in specific social contexts. Different cultures have different display rules, which are implicit or explicit norms about when and how different emotions should be expressed. These cultural norms are typically learned during childhood, through observation and through direct instruction, becoming internalized aspects of emotional responding that operate largely automatically. The existence of cultural variation highlights that while the basic neural and physiological mechanisms of emotion may be universal across humans, the development and expression of emotions occur within cultural contexts, which shape many aspects of emotional functioning. Language provides a window into cultural concepts of emotion, and languages differ substantially in the emotion words they contain and in how emotional experience is conceptualized and described. Some languages contain emotion words that have no direct equivalent in other languages, describing emotional states or experiences that are recognized and named within one cultural context, but may not be readily captured by the emotional vocabulary of other cultures. These linguistic differences do not necessarily mean that people in different cultures experience fundamentally different emotions, but they do suggest that cultural contexts influence how emotional experiences are attended to, categorized, and communicated. The study of emotion across cultural contexts requires careful attention to both universal features of human emotional functioning and the important ways in which culture shapes emotional experience, expression, and regulation. Theories of emotion have been proposed by researchers working in psychology, neuroscience, and related fields over more than a century. These theories emphasize different aspects of emotional functioning and propose different relationships between bodily changes, brain processes, and subjective experience. The James Lange theory, proposed independently by William James and Carl Lange in the late 19th century, emphasized the importance of bodily changes in generating emotional experience. According to this theory, emotional stimuli trigger bodily responses, such as changes in heart rate, breathing, and muscle tension. Emotional feelings arise from the perception of these bodily changes. In this view, we do not run because we are afraid, but rather we are afraid because we run with the bodily response preceding and causing the emotional feeling. The Cannon-Bard theory, proposed by Walter Cannon and Philip Bard in the early 20th century, challenged the James Lange theory on several grounds. Cannon argued that bodily changes occur too slowly to account for the rapid onset of emotional feelings, that different emotions are associated with similar patterns of bodily changes, which would make it difficult to distinguish between emotions based on bodily feedback alone, and that artificially inducing bodily changes does not reliably produce emotional feelings. The Cannon-Bard theory proposed instead that emotional stimuli are processed by the thalamus, which simultaneously sends signals to the cortex, producing emotional experience, and to the body, producing bodily changes. In this view, emotional experience and bodily changes occur in parallel rather than bodily changes causing emotional experience. The Schachter-Singer theory, also known as the two-factor theory of emotion, was proposed in the 1960s and emphasized the interaction between physiological arousal and cognitive appraisal in generating emotional experience. According to this theory, emotional experience requires both physiological arousal and a cognitive label or interpretation of that arousal. When individuals experience physiological arousal, they search their environment for an explanation of why they are aroused. The attribution they make determines which emotion they experience. If the same state of arousal occurs in different contexts, it could be interpreted as different emotions depending on the available contextual cues. This theory helped emphasize the importance of cognitive processes in emotion and the idea that emotional experience involves interpretation and not just the passive registration of bodily states. More recent theoretical approaches have built upon earlier theories while incorporating insights from neuroscience about the brain systems involved in emotion. Appraisal theories emphasize that emotions arise from evaluations or appraisals of situations in relation to an individual's goals and concerns. Different patterns of appraisal give rise to different emotions, with the specific emotion experienced depending on how the situation is evaluated along various dimensions, such as its relevance to goals, its implications for well-being, and whether it was caused by oneself or by external factors. These theories emphasize the cognitive components of emotion while also acknowledging the importance of bodily responses and neural processes. Construction theories propose that emotional experiences are constructed from more basic components, including bodily sensations, concepts, and contextual information, suggesting that emotions are not fixed biological responses, but are instead flexibly constructed in ways that depend on learning and cultural context. The process by which emotions gradually subside represents an important aspect of emotional functioning, involving multiple mechanisms operating at different timescales. When an emotional episode is first triggered, there is a rapid cascade of neural, hormonal, and physiological changes that constitute the emotional response. These changes reach peak intensity relatively quickly, typically within seconds to minutes of the triggering event. Following this peak, regulatory processes begin to act to gradually return the system to baseline, though the timescale of this return to baseline varies for different components of the emotional response. Neural activity patterns may shift relatively quickly as attention moves away from the emotional stimulus and as prefrontal regulatory mechanisms engage. Neurotransmitter and neuropeptide levels gradually return to baseline levels through mechanisms of reuptake, degradation, and changes in release rates. Hormonal changes, particularly increases in cortisol and other hormones released through the hypothalamic pituitary adrenal axis, take longer to resolve due to the slower time of hormonal signaling compared to neural signaling. Cortisol released during an emotional response may remain elevated for tens of minutes to hours before returning to baseline, depending on the intensity and duration of the emotional response and on individual characteristics. Cardiovascular changes also gradually resolve as sympathetic nervous system activation decreases and parasympathetic tone increases, bringing heart rate and blood pressure back toward resting levels. The subjective experience of emotion typically fades before all physiological changes have fully resolved, though residual physiological arousal can sometimes continue to influence experience and behavior even after the conscious emotional experience has substantially diminished. During the transition from waking to sleep, emotional processing undergoes gradual changes that parallel the changes in brain state associated with the descent into deeper sleep stages. As individuals relax and begin to disengage from active processing of environmental stimuli, activity in prefrontal cortex regions begins to decrease, while activity in some subcortical regions may initially increase before also declining. The emotional content of mental activity during the transition to sleep, including hypnagogic imagery and thought, may reflect this changing pattern as sleep deepens and individuals progress through lighter stages of non-rapid eye movement sleep toward deeper slow-wave sleep. Synchronized oscillatory activity comes to dominate. Cortical activity patterns and processing of emotional information becomes increasingly different from waking processing. In the deepest stages of non-rapid eye movement sleep, cortical neurons show synchronized patterns of activity consisting of alternating active and inactive periods, during which the neurons are largely unresponsive to input. During these periods of deep sleep, autonomic nervous system activity is at its most stable and reduced levels, with heart rate and blood pressure at their lowest points of the daily cycle. Breathing regular and slow. Hormonal secretion following patterns that support restorative processes. The subjective experience during these deep sleep periods, to the extent it occurs at all, is typically vague and fragmented rather than having the clear narrative and emotional structure of dreams that occur during rapid eye movement sleep. The progression through these sleep stages represents a gradual transition away from the patterns of neural and physiological activity associated with waking emotional processing toward very different patterns that support the restorative functions of sleep. The experience of emotions becomes increasingly distant and diminished as consciousness fades during the descent into deep sleep. The concerns, appraisals, and bodily sensations that constitute waking emotional experience gradually recede as brain activity patterns shift away from the configurations that support wakeful awareness. In the deepest stages of sleep, the neural systems involved in generating emotional responses show low levels of activity, and the patterns of communication between brain regions are fundamentally altered compared to the waking state. The prefrontal cortex, amygdala, hippocampus, and other structures show reduced activity and altered patterns of connectivity, creating a brain state in which emotional experience, as it occurs during waking, is largely absent. This progressive diminishment of emotional experience during the transition to deep sleep may serve important functions for emotional health and regulation. The daily cycle of engaging with emotional experiences during waking, followed by periods of deep sleep, during which emotional processing systems are relatively quiescent, may help prevent the accumulation of emotional arousal and may support processes of emotional memory consolidation and adaptation. The complete cycle of waking and sleeping, with its alternation between periods of active emotional engagement and periods of restorative sleep, during which emotional processing systems are relatively quiet, represents a fundamental rhythm of human emotional life that unfolds across each 24-hour period as sleep deepens and brain activity settles into the slow rhythmic patterns characteristic of deep sleep. Emotional experience fades into the background, giving way to other forms of neural processing that support the body's recovery and preparation for the next period of waking consciousness and emotional experience that will come with the following day. The neural mechanisms that support the gradual quieting of emotional systems during the transition to sleep and during sleep itself involve coordinated changes across multiple levels of brain organization, from individual neurons and synapses to large-scale networks of brain regions that communicate with one another through rhythmic patterns of activity as the brain transitions from wakefulness to sleep. There are systematic changes in the patterns of neurotransmitter release throughout the brain. Some neurotransmitter systems become less active, while others show different patterns of activity that are characteristic of sleep states. Neurons in the locus coeruleus, which release norepinephrine, are highly active during waking, particularly during attention and arousal, but gradually reduce their firing rates as sleep approaches, becoming nearly silent during deep non-rapid eye movement sleep. This reduction in norepinephrine signaling contributes to the overall decrease in arousal and the shift away from the alert, reactive state that characterizes waking consciousness. Similarly, neurons in the raphe nuclei, which produce and release serotonin, show changes in activity across the sleep-wake cycle, high during active waking, lower during quiet waking, and further reduced during sleep. Dopamine systems also show modifications, though changes vary across different brain regions and sleep stages. These systematic changes create a neurochemical environment during sleep that is fundamentally different from waking, and these differences contribute to altered patterns of information processing and emotional responding. The gradual shift in neurotransmitter activity as sleep approaches represents one of the mechanisms through which emotional reactivity is reduced and emotional processing systems are gradually quieted. Electrical activity patterns of the brain also undergo systematic changes during the transition to sleep and across different sleep stages. During alert waking, the electroencephalogram shows relatively low amplitude, high frequency activity, reflecting desynchronized activity of large populations of cortical neurons. As individuals become drowsy and begin to relax, alpha rhythm activity emerges. Rhythmic oscillations at approximately 8 to 12 cycles per second, particularly prominent over posterior regions. These alpha oscillations reflect a degree of synchronization as individuals disengage from active environmental processing and begin the transition towards sleep. As sleep onset occurs and individuals enter the first stage of non-rapid eye movement sleep, the electroencephalogram shows a mixture of frequencies with the emergence of theta activity, oscillations at approximately 4 to 7 cycles per second. During this light sleep stage, individuals are easily aroused and may not be fully aware that they have been asleep. Subjective experience during this stage, when it occurs at all, is fragmented and less coherent, reflecting the ongoing transition in brain state as sleep deepens into stage 2 non-rapid eye movement sleep. Distinctive patterns appear, including sleep spindles, brief bursts of rhythmic activity at 12 to 15 cycles per second, and K-complexes, large amplitude slow waves. These patterns reflect increasingly synchronized activity of cortical neurons and indicate a clear sleep state from which arousal is more difficult. With further deepening, slow wave activity increases, large amplitude oscillations at frequencies less than 4 cycles per second. These slow waves reflect synchronized transitions of large populations of cortical neurons between active and inactive states. During active phases, neurons may reactivate neural representations formed during prior waking experience, potentially supporting memory consolidation. During inactive phases, neurons are hyperpolarized, relatively unresponsive, representing periods where information processing is substantially reduced. The proportion of inactive phases increases as sleep deepens, reflecting the progressive reduction in overall neural responsiveness. Synchronization of neural activity during non-rapid eye movement sleep extends across large cortical regions. Slow waves propagate from frontal to posterior regions. This large-scale synchronization represents a fundamentally different mode of brain organization compared to waking, where brain regions process information relatively independently. The thalamus, a crucial relay for information, also shows altered activity patterns. Neurons enter a bursting mode, synchronized with cortical slow waves, further reducing responsiveness to external stimuli and diminishing information processing. Within this synchronized slow wave activity, brain regions involved in emotion show their own patterns of reduced activity. The amygdala, highly active during waking, particularly in response to threats or salient stimuli, shows reduced activity during non-rapid eye movement sleep. Connections between the amygdala and sensory cortices and prefrontal cortex show altered communication patterns. Reduced amygdala activity and altered connectivity mean that even if emotionally relevant information were processed during sleep, the response would be substantially different from waking responses. The prefrontal cortex, regions involved in conscious regulation of emotions and in integrating emotional information with goals, plans, and contextual knowledge, also show reduced activity during non-rapid eye movement sleep. Deactivation of the prefrontal cortex is particularly pronounced in regions involved in executive control and self-reflective processes. This contributes to the reduced capacity for volitional control and self-awareness that characterizes sleep states. Without the active engagement of prefrontal regulatory mechanisms, emotional processing during sleep, to the extent that it occurs, lacks the top-down modulation that shapes emotional experience. During waking, the combined reduction in activity of limbic regions and prefrontal regions creates a state where emotional processing is substantially diminished compared to waking consciousness. The hippocampus shows complex patterns of activity during sleep that differ from waking. During non-rapid eye movement sleep, the hippocampus shows sharp wave ripples, brief bursts of high-frequency oscillatory activity, during which neural representations formed during waking experience are reactivated in compressed form. These reactivation events play important roles in memory consolidation, allowing information encoded during waking to be gradually integrated into long-term memory stores. In cortical regions, when emotionally significant events are reactivated during these hippocampal sharp wave ripples, the reactivation may be accompanied by some degree of activity in emotional processing regions, though the nature of this activity and its relationship to emotional experience during sleep remains an area of ongoing investigation. Reactivation of emotional memories during sleep may contribute to processing and integration of emotional experiences, potentially helping to extract general patterns and meanings from specific emotional episodes. The hypothalamus, which coordinates hormonal and autonomic responses during emotional episodes, also shows altered patterns of activity during sleep. The circadian timing system, located in the suprachiasmatic nucleus, continues to function, maintaining the body's internal temporal organization, coordinating the timing of various physiological processes. Other hypothalamic nuclei, regulating arousal, metabolism, and hormonal secretion, show activity patterns appropriate for sleep states, supporting restorative processes while maintaining reduced levels of arousal and activation. The overall pattern supports reduced responsiveness to external demands and enhanced allocation of resources toward internal regulatory and restorative processes. The autonomic nervous system shows characteristic patterns during non-rapid eye movement sleep that differ substantially from waking patterns. Sympathetic nervous system activity, which is elevated during waking emotional arousal and supports increased cardiac output, elevated blood pressure, and mobilization of energy resources, shows reduced activity during non-rapid eye movement sleep. Parasympathetic nervous system activity, which supports restorative processes and reduces heart rate, supporting digestive and vegetative functions, shows relatively increased influence during non-rapid eye movement sleep. The balance between sympathetic and parasympathetic activity shifts substantially toward parasympathetic predominance during deep sleep, creating a physiological state characterized by reduced heart rate, reduced and stabilized blood pressure, regular breathing patterns, and enhanced activity of restorative processes throughout the body. The respiratory system shows particularly regular patterns during non-rapid eye movement sleep. Breathing rate and depth become more consistent and predictable than during waking. The variability in breathing that occurs during waking, where respiratory patterns are influenced by speech, physical activity, emotional states, and voluntary control, is largely absent during non-rapid eye movement sleep. Breathing is controlled primarily by automatic brainstem mechanisms that maintain appropriate levels of oxygen and carbon dioxide in the blood. This regular, automatic breathing contributes to the overall stability of physiological function during non-rapid eye movement sleep and reflects the reduced influence of emotional and cognitive factors on bodily processes. The gentle, rhythmic pattern of breathing during deep sleep represents one of the most noticeable physiological correlates of the reduced emotional and cognitive activity that characterizes this state. Cardiovascular function during non-rapid eye movement sleep shows reduced overall activation compared to waking. Heart rate typically reaches its lowest levels during the deepest stages of sleep. The variability in heart rate from beat to beat reflects the ongoing interaction between sympathetic and parasympathetic influences on the heart. This variability shows characteristic patterns during different sleep stages. During deep non-rapid eye movement sleep, heart rate variability shows prominent oscillations at very low frequencies, reflecting the influence of thermoregulatory and other slow physiological rhythms. Higher frequency variability related to respiratory rhythm remains present. The overall pattern of cardiovascular function during deep sleep reflects a state of reduced demand, with the heart working at a sustainable baseline level, appropriate for maintaining blood flow to tissues throughout the body while minimizing cardiovascular stress. Blood pressure follows a characteristic pattern across the sleep-wake cycle. Values during sleep are typically substantially lower than during waking. This nocturnal dipping of blood pressure normally involves reductions of 10 to 20% compared to daytime values. It reflects reduced sympathetic nervous system activity and altered regulation of vascular tone that occurs during sleep. The regular pattern of reduced blood pressure is thought to be important for cardiovascular health. Disruptions of this pattern, with absent or reduced nocturnal dipping, have been associated with increased cardiovascular risk. The reduced blood pressure during sleep reflects the overall reduction in physiological activation and arousal that characterizes sleep states and contributes to the restorative functions of sleep. For the cardiovascular system, the endocrine system shows complex patterns of hormonal secretion that vary across the sleep-wake cycle in ways that support different physiological needs. During waking and during sleeping, growth hormone secretion shows a prominent surge during the early part of the night, typically in association with the first episode of deep slow-wave sleep. This pattern of growth hormone release supports tissue growth and and repair processes that occur preferentially during sleep. Cortisol secretion follows a circadian pattern, with levels lowest during the early night and rising during the later part of the sleep period, reaching peak levels around the time of usual awakening. This pattern of cortisol secretion helps prepare the body for the metabolic and behavioral demands of the upcoming waking period. Thyroid stimulating hormone shows maximal secretion during the night, supporting thyroid hormone production, while prolactin levels rise. During sleep, these various patterns of hormonal secretion create an endocrine environment during sleep that differs substantially from waking and supports the physiological processes that occur preferentially during sleep. The regulation of body temperature also shows characteristic patterns. During sleep, core body temperature typically decreases during the night and reaches its lowest point during the early morning hours. This nocturnal decrease in body temperature reflects reduced metabolic heat production and is coordinated with the circadian timing system and with sleep-wake patterns. The decrease in body temperature during sleep may support some of the restorative functions of sleep and reflects the overall reduction in physiological activation, the sensation of temperature, and the body's thermoregulatory responses continue to operate during sleep, though with altered sensitivity compared to waking. Disruptions of thermal comfort can lead to sleep fragmentation and lighter sleep stages. The gradual decline in body temperature across the night parallels the progressive deepening of sleep and the increasing dominance of slow-wave activity during the first portion of the night. The immune system shows important interactions with sleep. Various immune functions show enhancement during sleep periods. Certain cytokines that play roles in immune responses show increased production during sleep. There is evidence that sleep supports immune memory formation and the development of protective immune responses. Conversely, sleep disruption or sleep deprivation can impair immune function, potentially increasing vulnerability to infections and reducing the effectiveness of immune responses to vaccines and other challenges. The relationship between sleep and immune function represents another example Of the many ways in which sleep supports bodily regulation and maintenance, with reduced behavioral and cognitive activity during sleep, allowing resources to be allocated toward immune surveillance and defensive functions. The processing of emotional memories during sleep appears to involve the selective reactivation and strengthening of memory traces. Emotionally arousing memories show preferential consolidation compared to neutral memories. Studies examining memory for emotional versus neutral material have generally found that sleep, particularly sleep-rich and rapid eye movement sleep, or sleep that includes adequate amounts of slow-wave sleep, preferentially benefits memory for emotional material. This selective benefit for emotional memories may occur through mechanisms involving the reactivation of emotional memory traces, potentially accompanied by patterns of neural activity in the amygdala and other emotional processing regions. Though these neural responses during sleep differ substantially from the responses that occur during waking emotional experience, the strengthening of emotional memories during sleep may help individuals learn from emotional experiences and adjust future behavior accordingly. Processing emotional memories in the altered neurochemical and physiological context of sleep may contribute to emotional adaptation and regulation. Some theoretical perspectives suggest that the processing of emotional memories during sleep may help to dissociate the emotional intensity of memories from their factual content, allowing individuals to retain information about what happened during emotional events while reducing the physiological and subjective emotional response that occurs when those memories are later recalled. According to this view, the reactivation of emotional memories during sleep, particularly during rapid eye movement sleep, when emotional processing regions such as the amygdala are active, but stress hormone levels are low, may allow for a gradual weakening of the emotional components of memories, while preserving the informational content. This process, occurring over repeated cycles of sleep across multiple nights, may contribute to emotional recovery following difficult experiences, and may help explain why adequate sleep appears to be important for emotional well-being and resilience. The transition from non-rapid eye movement sleep to rapid eye movement sleep involves systematic changes in brain activity patterns and physiological regulation as rapid eye movement sleep begins. The synchronized slow wave activity that characterizes deep non-rapid eye movement sleep gives way to faster frequency, lower amplitude electrical activity that resembles in some ways the pattern seen during waking. However, despite these similarities in cortical electrical activity patterns, rapid eye movement sleep is a distinct state with its own characteristic features. During rapid eye movement sleep, muscle tone throughout the body is actively inhibited, producing a state of muscular atonia, except for the diaphragm and other respiratory muscles. The extraocular muscles that produce the rapid eye movements that give this sleep stage its name, and a few other exceptions. This muscle atonia prevents the physical acting out of dream content and represents one of the most distinctive features of rapid eye movement sleep. The pattern of brain activation during rapid eye movement sleep includes high levels of activity in limbic structures, including the amygdala and the hippocampus, while prefrontal cortex regions, particularly dorsolateral prefrontal cortex, show reduced activity compared to waking. This pattern of limbic activation, in the absence of full prefrontal engagement, has been proposed to contribute to the often emotionally intense and sometimes bizarre quality of dreams that occur during rapid eye movement sleep. Visual cortical regions show activation during rapid eye movement sleep, potentially contributing to the vivid visual imagery that characterizes many dreams, while motor cortex regions also show activity, though the physical expression of this activity is blocked by the muscle atonia that characterizes this state. The overall pattern of brain activity during rapid eye movement sleep supports the generation of complex mental experiences that can include rich emotional content, though these experiences differ in various ways, from waking consciousness, autonomic nervous system regulation during rapid eye movement sleep shows greater variability compared to non-rapid eye movement sleep, with heart rate and blood pressure showing moment-to-moment fluctuations. that may correspond to dream content. Respiratory patterns during rapid eye movement sleep are less regular than during non-rapid eye movement sleep, showing variability in rate and depth. These phasic changes in autonomic function during rapid eye movement sleep reflect the greater activation of limbic and other brain regions during this state, and suggest that emotional processing during rapid eye movement sleep dreams may be accompanied by corresponding physiological responses, though these responses are generally less intense than would occur during waking emotional experiences. The overall level of autonomic activation during rapid eye movement sleep is intermediate between the low stable levels seen during deep non-rapid eye movement sleep and the variable, often elevated levels seen during active waking. The neurochemical environment during rapid eye movement sleep differs substantially from both waking and non-rapid eye movement sleep. Acetylcholine levels in many brain regions are high during rapid eye movement sleep, similar to or even exceeding waking levels. This cholinergic activation is thought to contribute to the cortical activation patterns and the generation of dreams during this state. In contrast, norepinephrine and serotonin release are minimal during rapid eye movement sleep, creating a neurochemical context that differs substantially from waking. This absence of norepinephrine and serotonin during rapid eye movement sleep may have important implications for emotional memory processing, as these neurotransmitters play roles in stress responses and emotional reactivity during waking. The processing of emotional memories during rapid eye movement sleep, in the absence of these neurotransmitters, may allow for emotional learning and adaptation to occur in a context where stress responses are minimized. The cycling between non-rapid eye movement and rapid eye movement sleep continues throughout the night, with the brain progressing through a series of sleep cycles, each lasting approximately 90 minutes. Early in the night, sleep cycles tend to contain more deep slow wave sleep and relatively brief episodes of rapid eye movement sleep. As the night progresses, the amount of slow wave sleep in each cycle decreases, while rapid eye movement sleep episodes become longer and more intense. This ultradian rhythm of sleep stages creates a pattern where the deepest sleep and lowest levels of arousal and emotional processing occur early in the night, while the later part of the sleep period contains increasing amounts of rapid eye movement sleep with its associated dreaming and emotional memory processing. The complete pattern of sleep across the night represents a complex temporal organization of different types of neural processing, physiological regulation, and restorative functions. The functions of this cycling between different sleep stages and the specific roles of non-rapid eye movement and rapid eye movement sleep remain topics of active research and theoretical debate. Both sleep stages appear to contribute to various aspects of memory consolidation, with non-rapid eye movement sleep particularly important for certain types of declarative memory, while rapid eye movement sleep plays roles in procedural memory and possibly in emotional memory processing and integration. The presence of both sleep stages across mammals suggests that both serve important functions that have been conserved through evolution. The specific patterns of brain activity, neurochemical environment, and physiological regulation that characterize each sleep stage create different contexts for information processing and memory consolidation, suggesting that the cycling between these states allows for complementary forms of processing that together support learning, memory, emotional regulation, and other functions. The importance of sleep for emotional functioning is underscored by the effects of sleep deprivation on emotional responses and regulation. Even modest amounts of sleep restriction, such as reducing sleep time from 8 hours to 5 or 6 hours per night, have been found to produce measurable changes in emotional reactivity and regulation. Studies using neuroimaging have shown that sleep deprivation is associated with increased amygdala reactivity to negative emotional stimuli, while at the same time, the functional connectivity between amygdala and prefrontal cortex regions involved in regulation is reduced. This pattern suggests that sleep deprivation may lead to emotional processing that is more reactive and less effectively regulated, potentially contributing to emotional difficulties. Behavioral studies have confirmed that sleep-deprived individuals show increased emotional reactivity, particularly to negative stimuli, and report more negative mood states. The mechanisms through which sleep deprivation affects emotional processing likely involve multiple pathways. The extended period of waking during sleep deprivation may lead to accumulation of adenosine and other metabolic byproducts in the brain that influence neural function. Sleep deprivation affects various neurotransmitter systems, potentially altering the balance of dopamine, serotonin, and other signaling molecules in ways that influence emotional processing. The absence of the specific forms of neural processing that occur during sleep, including memory consolidation and synaptic homeostasis, may lead to alterations in neural circuit function that affect emotional responses. The disruption of circadian rhythms that can occur with chronic sleep restriction may further contribute to emotional difficulties through effects on hormonal regulation and other circadian-controlled processes. The accumulation of these various effects explains why adequate sleep is important for maintaining stable emotional functioning. Recovery from sleep deprivation typically involves sleep that is enriched in slow-wave sleep, particularly during the early part of the recovery sleep period, as the brain appears to compensate for the lost deep sleep. This recovery sleep shows increased intensity of slow-wave activity compared to normal sleep, reflecting enhanced synchronization of cortical neurons. Following recovery sleep, emotional reactivity and regulation typically improve, though the time required for complete normalization may extend beyond a single night, depending on the extent and duration of prior sleep deprivation. The restoration of normal emotional functioning with recovery sleep underscores the active role that sleep plays in maintaining emotional regulation, rather than sleep being simply a passive period of inactivity. The relationship between sleep and emotional functioning operates bidirectionally, with sleep affecting emotional responses during subsequent waking periods, and with emotional experiences during waking affecting subsequent sleep. Emotionally arousing experiences during the day can influence sleep onset and sleep quality, sometimes making it more difficult to fall asleep or leading to more fragmented sleep with more frequent awakenings. The processing of emotional experiences during waking continues into the sleep period, where emotional memories may be reactivated and consolidated, potentially influencing dream content and the patterns of neural activity during sleep. This ongoing interaction between waking emotional experience and sleep creates a continuous cycle where emotional processing during waking influences sleep. Sleep influences emotional functioning during the next waking period, which then influences the following night's sleep, and so on, across days and weeks and longer timescales. The circadian timing system, which organizes many aspects of physiology and behavior across the 24-hour day, also influences emotional functioning. There is evidence for circadian variations in mood and emotional reactivity, with many individuals reporting particular times of day when they feel most positive or when they are most emotionally reactive. These circadian variations in emotional experience may reflect underlying rhythms in neurotransmitter function, hormonal secretion, or other physiological processes that influence emotional processing systems. The circadian system and sleep-wake regulation interact closely with both systems contributing to the temporal organization of emotional functioning across the day and night. Disruptions of circadian rhythms, such as can occur with shift work, jet lag, or irregular sleep schedules, can contribute to emotional difficulties, likely through combined effects on sleep quality and circadian organization of emotional processing systems. The gradual quieting of emotional systems that occurs during the transition to sleep and during sleep itself represents a natural, recurring process that allows the brain and body to shift away from the aroused, reactive state that characterizes waking consciousness towards states that support different functions, including rest, restoration, and particular forms of memory processing. This daily cycling between waking emotional engagement and sleeping emotional quietude represents a fundamental rhythm of human life that has been shaped by evolution and that serves important functions for maintaining emotional health and adaptive functioning. The neural mechanisms that support this cycling involve coordinated changes across multiple brain regions, neurotransmitter systems, and physiological regulatory mechanisms, all working together to create the characteristic patterns of emotional experience and processing that occur during waking and sleep states. Now, your mind is calm, your heart is steady, your feelings can drift like leaves on a quiet stream, slowly, gently, peacefully. All that you felt today, all that stirred within you, is acknowledged, accepted, and allowed to rest. There is nothing else you need to do, nothing to control, nothing to resist. Everything is gently settling. Relax. Breathe. Let your emotions flow naturally. Sleep is here. Sleep is safe. Sleep is gentle. Your mind, your heart, your body, all rest now, peacefully, softly, completely. Good night, beautiful soul. Sleep and dream, and let your inner world quietly, softly heal and restore through the night.